But I wanted to thank all of you for your hard work. I've been doing Earthwatch since 1995, and it's been very, very rewarding. We have just, as an extended group, I think there are about 1,200 volunteers now who've been part of the Caterpillars project. We've created, I think, an awesome data set and really great memories and just, I think, good connections and good things have come from this work. So I really appreciate your interest and your involvement and the help, and I know that Nick appreciates this. This was good training for Nick. It's also really good data for Nick. He's a first-year graduate student, and I'm certain that he'll do a lot of really good things with the data that you all have helped him collect here at the beginning. So before I show you some of the things, I kind of wanted to see what you all thought we're going to do with this. So we got caterpillars in bags, we have pictures, we have some insects and alcohol, we've got some extracts that you made in the lab, we have some plot data, we have an Excel spreadsheet with a bunch of data in it. What are we going to do with all this stuff? Because those aren't the products, those are works in progress. But one thing that comes to mind whenever we get this feedback is that this site's a little different, and our Arizona site too, because we do a lot of beating. But at our Costa Rica site, for example, there's a lot of broadleaf things, and you turn over leaves all the time, a lot, and it's a lot harder to find caterpillars there. It's kind of like, it's worse than the juniper. Worse than a bad day in the juniper. We've had volunteers come back after a full day and they didn't get a single caterpillar. They look so dejected. We have to help them, find the right leaves to turn over. But a lot of them say that whenever they go to a forest, they'll get emails that say, I can't walk through a forest without turning leaves over. So since you didn't do a lot of that, I'll say that if you're out with students for a walk or something, turn some leaves over and you might find a cool caterpillar, and then you can sort of relate it back to this. And some of the ideas that you've just mentioned and how you can link this back to the classroom. But then also some of the science, because I'm hoping that some of the science will make sense and that you'll even want to look at some of our papers, which I know are difficult to read, but there's still some information you can get from those papers. And we do try as hard as we can to interface with more popular literature. So as Lakeisha mentioned, a book would be really handy, and so our collaborator, Dave Wagner, who wrote the Eastern Caterpillars Guide, he's coming to our Arizona sites every year now, because he wants to write the Western Caterpillars Guide. He's going to come to this site. He's going to come this year, but he's just two. He's got another book coming out on the owlet caterpillars. But that is not, I mostly publish in the primary literature, but we do interface with people like WAGs so that they can write these books for the popular press. And that book was really popular, actually. Eastern Caterpillars, tons of people bought it. It did like academics never do this. It did like a book signing. So this is what we did. I'm not playing favorites, I just took a few pictures here. So it's, we collected over 525 individual caterpillars. This group did. The group before you, the girls collected another several hundred caterpillars. So we almost got 1,000 as two teams, which is good for this area. It's really good. And it's more than what we typically get in Costa Rica, although the diversity is much higher. The number of families is higher in Costa Rica. I was also impressed with the schedule that Mariah put up and some of the procedures that are important to follow when you're doing zoo. We collected caterpillars in 10 different families, about 80 different species, and we also did a lot of other things. Right, you all learned hopefully a little bit about chemistry and natural products chemistry, which is really, although we don't do a ton of it with the Earthwatch groups, it's a very important part of the work we do. And the interactions between plants, caterpillars, and parasitoids are mediated by chemistry, as hopefully Angela pointed out, or I know that she pointed it out in her talk. Hopefully it's pretty clear that chemistry is a really important part of these interactions. And then there's also applications, as I'll mention in a minute, to the work we do to more applied things such as regulation of mosquito populations. So all these trophic interactions aren't just limited to plant caterpillars and parasitoids. They could be, plants and mammalian herbivores, ungulates, and their vertebrate predators. In fact, there was a paper in Science not long ago that we worked with vertebrates in the Serengeti, and they cited some of our work because it's the same kind of thing, these trophic interactions. Right? They were interested in how predators affect plant biomass and plant chemistry. Right. Right. We also focused on about a dozen plant species, which helps us because you all are gone now, and so our crew here has to take care of all of your babies that we collected. And they're in good hands. And part of the reason why it's going to be not terribly hard is because we don't have too many plants to go collect when we need to give them fresh food. Right? Mostly it's juniper, and everybody recognizes the one on the left. Cenosis and got it. Awesome.
With a W.
Yeah, that's right. And after Racy too. So great job, everyone. We also had some time for, well, you all had some time for relaxation up at the lake. Lake Tahoe is a pretty nice place. It's too bad you couldn't go to Pyramid Lake as well, but it's the time here is too short. Okay, so here's the families that we collected. Parallid Gallichiids, Tortricids I didn't write up there. I don't know why it's that there. There's Nodontids, Archaeids, and I think Nymphalidus. Somebody wrote that in the database. It's been described to me, but I haven't seen an image yet. So I'm assuming that's correct. Okay, so here's the examples of what you collected. The top one actually is from a previous team. So this is everybody remember this family? These are shelter builders. It's all poopy inside their shelters. The parallid. These are parallids, and this is a closely related family in the Paraloides superfamily. Do you remember this one?
There's a Gallichiid.
That's the next one. This is Tortricids. Tortricids, and these are Gallichiids here. We think, we don't know for sure. We'll have to see what comes out. But those are the Gallichiids that you got on Juniper. You got, does anyone know what the top one is? Yeah.
You know that one.
The bottom one, that's in the act of pooping. I didn't get his permission before I took that picture, but that we didn't collect. I actually thought we did have a Pierret at one point, but it turned out to be a sawfly, so. Very big bummer, so we didn't get Pierrets. The only butterflies we got were Lycaenids and then Nymphalids. We didn't get any Hesperiids. So none of these are from Markman. We didn't get any Papilionids. It says in the database Nymphalid, but I didn't see Nymphalid. Okay. Is there an image of it?
Today.
Okay. All right, so you got a Nymphalid. So Nymphalids typically look like those top two pictures there.
The one guy.
We
Yeah,
have one of those. It was brown and orange.
Okay.
Yeah.
Sounds like an Nymphalid. What was it? Do you know? You should know.
It was feeding.
You guys didn't know?
I was thinking
It's
it's
a wax current.
Wax current is what
Yeah.
it was.
Huh.
There's so many checker spots up
It
there
was
that
probably,
it makes me think, but it's big for a checker. It's a big caterpillar.
it could be, Spierea or something like that.
Geo.
And then, of course, everybody knows this family. Yeah, geometry. These are all yours. These are all from your bags. They're here in good hands, as I said. So those are some of your geometrians. Any site, every single site, this is our biggest family. It's incredibly diverse. The adults are diverse as well. They're really attractive caterpillars, I think, and they're ecologically relevant. They're dominant. They eat they turn a lot of plant into caterpillar, and then other animals can turn caterpillar into other things. So they support big, diverse communities. I didn't get a picture of the Nodonta that you all got, but this is what Nodontas look like. These are some Nodontas from Arizona. And I did get it sounds like you may have got an Archaeid, but I wasn't sure. There are a few things in the database that said unknown, but this is what Archaeids look like. And this is an Archaeid that eats Doug fir. These are your Noctuids. These are a few of your Noctuids. The one on the bottom there is Spinothera. And that is the most diverse family, but it's not the most abundant in any in most forests. It's not really the most abundant. And here's just an example of diversity of Noctuids from North America. These are from both here, this site, and also the Arizona site.
So was it hard for you to determine that these are all the same, or you just counted the legs or something like that?
But
Because
they're
they
all,
look very different. Like when you were looking for them, how did you know that all of these were the same family?
well, Noctuidy that's a good question. I mean, Noctuids, there's a bunch of subfamilies that if you know the subfamilies, you'll know. So for example, the Kataka lines always have reduced number of prolegs and look a little bit like geometrids. There's the acronictines that are hairy. There are a bunch of subfamilies or even genera that you just will recognize right away. the way to tell noctuids is based on the arrangement of their crochets on their forelegs, which you can't tell from a picture. The other way is that you just know. So somebody like Dave Wagner or another guy who's a really good natural historian, Mike Singer from Connecticut, they don't necessarily know all the characters. They've just seen thousands of species of caterpillars and they're like, oh yeah, that's that, and that happens to be a noctuid. And in fact, a lot of people in the field, that's the way they are. They don't actually know why something's a noctuid. They just know all the noctuids. They have that kind of mind. They're like, oh, that's that, that's that, that's that. Like Nick's going to meet some of those guys when he goes to Arizona. They're these crazy, crazy taxonomist types that just know all these names, but they don't really know the relationships. Sometimes they don't even know what family they're in. They just know all the Latin binomials. That's okay. So there are some conclusions that we will be able to draw eventually from these data, but like I said, this is a long-term process. Right? So this was when Cam was this age. This was about 5 years ago in Ecuador. We were collecting data that I'm just now putting into some models and using, you know, to address some questions. And just to remind you, some of the questions that our lab is interested in, and we're never going to really get definitive answers to these, but we're going to get answers to smaller questions that are related to these big questions, like how many species there are in the world. So for example, that first question, you know, as we nail down, so within the Noctuidae, for example, the owlet moths, as we figure out more and more about their feeding relationships with plants, and as we figure out how specialized they are at different sites, we can get better and better and more refined estimates of how many species of Lepidoptera there are. And that will fit with other people's ideas of how many species of other animals there are, and we can, you know, help answer this question. And you know, for a lot of people this is an esoteric question. They might not really care how many species there are, but again, all the smaller questions that are related to this can have applied or conservation value, such as what type of ecosystem supports the most number of species with greatest biodiversity. And so these are the kinds of things that we'll use the data to address. You know, like I said, I wanted to keep this talk short, so I'm not going to go into all of, you know, how we would address all these questions, but I'll just say that we will try to contribute to these. It's a little blurry. Let's see. Have all the ones been blurry? That's better. Yeah. The other thing that is a major product that one of you pointed out is that we really want to contribute to the natural history of the Great Basin in the Sierra Nevada. Right? So a lot of these caterpillars, still at this point, most of the caterpillars you collected have not been described. These immature stages haven't been described. So there are a few aspects of this. So for example, this was a new species of moth that we discovered by collecting a caterpillar. Right, so this moth got a name, and that's an important part of natural history. We knew that this thing existed at this particular research site. We also described the immature stages. We even had images of the eggs. So this was the egg stage. And we reared out parasitoids. This is a Burkhanid parasitoid. So we basically, at all of our sites, are starting to resolve these really complex food webs. Right? And sometimes it's going to be focused on, you got to start somewhere. So for here, one of the things we're doing is we have focal plant taxa. And we're going to try to get the food webs resolved for juniper and for ceanothus and for a couple of other focal plant genera. And that's what we do at all of our sites. So for example, in Ecuador and Costa Rica, we focus on piper, which is a pretty big chunk to try to work with. There's 2,000 species of piper, but it's a good starting point. So the other thing that we're producing is natural history. And if you want to know this kind of stuff, you can go to caterpillars.org. And eventually, the pictures that you took and that others who came here in Earthwatch will end up on caterpillars.org. So yesterday, Judith and Rakisha were working on taking the images that you took and making them web ready. So they were cropping them and making them smaller size, so that when we make the web pages, once we figure out what species it is, which could take a while, we make the web page, we put that image in there, and then people have, if you go to caterpillars.org, I would recommend, for example, going to the Arctiids of Ecuador, and you can see some really nice pages that are very thorough. We have the adults, the immatures, all the food plants that they eat. We've got the natural history provided for people. Again, this will also help people like Dave Wagner who are working on books like the Western Caterpillars of North America.
Is that the moth that's on your door?
No, this is much smaller. This is a dioptine notodonid. It's like the one that sort of like the one I have tattooed on my leg. extended
yours,
phosphates.
right?
No, this is a different one,
Ah.
yeah. There's another new species, but it's I think he called this Yana yakuensis. He named it after the research site. Yanayaku. Of course, the name of this project is Climate Change in Caterpillars, and one of the big questions that we will continue to try to address is how things like extreme weather events, which is one of the things I mentioned that we've already published, but we're still really looking at, especially at our Louisiana site, and here as well, where we can get really variable weather patterns. We want to see how it does things like disrupt parasitism. So if you remember, as variation and year-to-year variation in rainfall increases, parasitism decreases. That's a really broad general pattern. There's a lot of other hypotheses that we can address. We can do here. We could do experiments like we do in Louisiana, where we do some flooding to figure out exactly which parasitoids are affected the most. One of the things I'm focusing on now with all the Earthwatch data across all the sites is trying to figure out how the Burkhanid in particular are affected, the Burkhanidae family. Right, looking to see if there are certain subfamilies that might do better under extreme weather or under increasing CO2 or temperature. Right, so this is a focus that we still have, and we can still use the data that you helped collect to address more of these questions. Then there are a ton of applied issues, one of which was you had first-hand experience with the mosquitoes and predator communities that can regulate mosquitoes. One of the things we've studied a lot is we've created models using our data, our Earthwatch data, to predict whether or not biological control can be successful. Right? When I first started doing research as a graduate student, I met a crusty old biologist who studied insects, and he's what entomologists call a nozzlehead. He's very interested in killing insects. And actually, those who are interested in killing insects have taught us a lot about insects. Because to figure out how to kill them, you have to know a lot about them. And when I mentioned I wanted to study biocontrol, he said, he's like, I can tell you everything you need to know about it. It doesn't work. And the fact is that a lot of biological control programs have been failures. And on the other hand, though, a lot have been successes. And what we were able to do with our Earthwatch data is predict whether or not biocontrol would be a failure. And that kind of information can make biological control more successful. So a really simple example is if you have a specialist herbivore pest, don't use predators to control it because they don't like specialists. That's something we figured out with Earthwatch data and published a bunch of papers on. And then we put it in our model, and it's really clear. It's like all these failures you could have prevented if you just used a little bit of natural history information, right? If you knew, and so the more parasitism data we have, the more we'll know that, okay, meteorines don't like brightly colored caterpillars. Don't use them in biocontrol to test what your pest looks like. Or ants don't like hairy caterpillars. You can't enhance your ant community to try to deal with a hairy caterpillar pest, and so on. So that's the kind of applied thing that can come from this kind of, seemingly esoteric natural history or addressing basic ecological questions. And we'll continue to do that with our data as well. So that's another goal. And the point of this is that we have a lot of applied goals as well as addressing those really basic research questions. And then the last thing is that a goal of our lab and sort of a career goal for me is to really figure out this idea of how we quantify and how we understand interaction diversity. Right? When I think of places that have been lost, for forests or patches of desert, habitat loss, there are a lot of species that are lost, but I also think of stories like the piper plant we found that's got these beetles living in it that kill these ants and that are really, really specialized in very small regions. And those kind of stories of interactions are really important. It's kind of like books in the library that if you were to burn down all the libraries, you lose all those stories. They're gone forever. That's what you lose when you lose forests. You lose these interactions. And I agree with John Thompson that if you don't maintain this kind of interaction biodiversity, whether they're pollinators or the trophic interactions we study or competitive interactions, you're going to lose biodiversity. These interactions are what maintain the diversity that we see. So this is really the ultimate goal, is that we document interaction diversity and that we come up with a method, and the plot method is one of them, it's not the only one, for documenting this kind of interaction diversity. So that's that's the final product that will take a very, very long time. Maybe 20, Cam will be an adult when we finally have some really good handles on how interaction diversity behaves. So the other thing that I like to mention is that the products are not just, from the Earthwatch data, are not just papers and journals. There are a lot of things that Earthwatchers have produced and contributed to, including the papers and peer-reviewed journals. And this is what academics care about the most, and not just so that they can have a bunch of ticks on how many papers they have, it's because this is the way science moves forward. You have to publish in peer-reviewed journals, and that eventually makes its way into textbooks and into popular knowledge. Science is a way of knowing, and the way we currently know things is to publish what we found in scientific journals. The data has contributed to several books, thousands of species websites. I really encourage you to explore caterpillars.org, and maybe your students could use it for something. Many teachers have made very impressive websites. I've really enjoyed looking at teacher web pages, so if you have websites, let us know and we'll check them out, put links to them even. A bunch of high school students from SCAP are now biology majors in college. You actually met the former SCAP volunteer who's going to start graduate school next year. Mariah was a SCAP volunteer. Former regular Earthwatch volunteers, many of them got PhDs in tropical entomology or ecology. And for me, one of the really important things at our sites in Latin America, in Costa Rica and Ecuador, is that we've trained what some term para-ecologists, and so these are people like Umberto there in the picture who never finished grade school and worked in the banana plantations and then worked in the taller, the workshop at the station, and then he started working on our caterpillar project, and now he's the region's expert on caterpillars. Nobody knows as much about caterpillars and their host planets as Beto does. And it's true for our crew in Ecuador as well. And ultimately for those sites, I mean, I know I travel a lot, but I don't like traveling very much. And I would love those sites to just sort of run themselves, and I can, not be involved in them anymore, or not be involved as intensively. And the people who are running them can have ownership and then hire their own colleagues to work with them. So that's another product, I think. There's also some really nice children's books that have come out of the Earthwatch, various Earthwatch teams.